Hello everyone. Today I will talk about endometrial organoids and associated diseases. I'm Yan Lei from the Reproductive Hospital affiliated to Shandong University. Now this is the outline. I will talk about this issue from the three parts: human endometrium, introduction of organoids, and endometrial organoids. Firstly, endometrium still remains poorly understood today. It is organized into two functional distinct areas: superficial functionalis and the deeper bacillus. Menstruation, or regular shedding of the functionalis, is the most distinctive feature of human female reproduction. Endometrium is the primary target organ for ovarian hormones. With monthly remodeling, the functionalis epithelia proliferate massively in response to estrogen in the proliferative phase. During the secretory phase, differentiation and changes in cellular morphology owing to the increase in progesterone. The result is either embryo implantation, if the released oocyte is fertilized, or menstruation at the end of the menstrual cycle. This picture shows the proposed endometrial epithelium regeneration. Figure A shows a 2D schematic of the full-thickness endometrium, highlighting the potential locations of epithelial stem or progenitor cells within the different zones of the bacillus. Figure B shows the maintenance of the luminal epithelium by resident stem or progenitor cells following the daily shedding. Desquamation events and blastocyst implantation. The mycelium-like configuration of the stem's research bacillus will allow rapid replenishment of the lost bacillus glands by horizontal spread in a way that would not be possible with a bland singular glandular conformation. Menstrual cycle is shared with only a few species, including apes, bats, and elephant. It does not occur in any of the standard model organisms that undergo sexual reproduction, such as the mouse, zebrafish, or fruit fly. So how will we research or study benign endometrial diseases such as endometriosis and adenomyosis? Recently, 3D histologic reconstruction forming organoids demonstrated a much more complex architectural organization. So what is organoid? Organoid refers to cells growing in defined 3D environment in vitro to form many clusters of cells that self-organize and differentiate into functional cell types, recapitulating the structure and function of an organ in vivo. Different tissue-specific 3D organoids like kidney, lung, and brain, shown here. Importantly, patient-derived cancer organoids have been researched today. This is a brief history of organoids. Back in 1907, Wilson described the first attempt of in vitro organism regeneration. Few decades later, several groups performed dissociation reaggregation experiments to generate different types of organoids from dissociated amphibian pronephros and chick embryos. In 2009, a landmark study from Hans first reported on establishing a 3D organoid culture derived from a single AIC, which set the scene for many subsequent organoid works in other systems, including stomach, liver, pancreas, lung, and kidney, and neurorectums, using AIC or from human pluripotent stem cells. Now, three methods are frequently used in organoid derivation. Firstly, the physical features of the cultural environment are important to organoid development. The main types of cultural environments are shown here, together with examples of organoids that rely on them. Secondly, the formation of a specific organoid depends on different degrees of system autonomous endogenous signals and exogenously supplied signals. Mouse optic curve formation depends largely on endogenous signals, like the left. Derivation of kidney organoids initially relies on exogenous signals that induce the ureteric epithelium and the metanephric mesenchyme from human PSC. Subsequent self-organization steps rely on endogenous signals, like the center. The supply of specific exogenous signals is continuously required for HPSC-derived gastric organoids, like the right. The necessity for exogenous signals likely substitutes for missing cells types that are responsible for the production of these signals during in vivo organogenesis. Thirdly, organoids can be formed from cell populations with different starting conditions. Examples of an organoid protocol starting from a single cell or a homogeneous multicellular ensemble such as optic organoids, and a heterogeneous co-culture of different cell types such as liver organoids are shown here. The initial conditions are aligned with different steps of organoid development. Nowadays, induced pluripotent stem cells, that is iPS, and adult stem cells have been widespread utilized in establishing organoids. Speak over here. Has endometrial organoids been established today? And how is the progress and potential in organoid research in the future? For the first question, the answer is yes. Endometrial organoids have been created. This picture showed that the development of organoids from mouse endometrium and reproduce the phenotype of the epithelium. Time-lapse recording of organoid development from mouse endometrium and endometrial organoids reproduce endometrial epithelium phenotype by immunohistochemistry, staining, and transmission electron microscope. Reveals the presence of microvilli in endometrial organoids at the luminal site. This picture showed that the development of organoids from human endometrium and reproduce the phenotype of the epithelium. Similar results with the mouse endometrial organoid. Most of all, human endometrial organoids show physiological responses to hormones and reproduce the menstrual cycle. Recent studies confirmed that the human EMOs indeed highly mimic the mechanically sensitive channels in the endometrial epithelium cells, including piezo-1 channel. To summarize, types of endometrial organoids including monocellular endometrial organoids and multicellular endometrial organoids. The characterization of endometrial organoids includes the following: express specific marker of the original tissue, and show physical responses to hormone treatment, and recapitulate genomic signatures of tissue of origin. The application of endometrial organoids, including endometrial development, endometrium embryo crosstalk, which is a model for studying extracellular vesicles in endometrial biology. Then let's review the organoid models of endometriosis. Long-term expandable organoids from patient-matched eutopic endometrium and ectopic lesions shown here. Compared with organoids from eutopic and control endometrium, ectopic organoids also exhibited ERF and PGR expression, microvilli and cilia directed towards the lumen, and secreted mucus into the lumen. Endometriotic organoid biobanks will be valuable in deciphering disease-specific pathogenesis. Organoids from samples of endometrial cancer and normal adjacent endometrium from postmenopausal women, supporting the idea that this model recapitulates the histological organization and phenotypes of the endometrial cancer. Interestingly, these organoids accurately captured the heterogeneity and mutational landscape of the tumor, recapitulated disease phenotype, and display patient-specific drug responses. These endometrial cancer-derived organoids may be used in the future to build a biobank for drug screening and investigating the mutational changes, and could possibly replace the use of patient-derived exografts, mouse models, and decrease the overall costs associated with in vivo models. Scaffold-free EMOs comprising of endometrial and stromal cells from human endometrial tissues, shows here. These organoids exhibited functional characteristics of the normal endometrium in response to follicle phase estrogen and testosterone. Treatment with human excess androgens mimicking PCOS, increased cell proliferation and dysregulation genes associated with proliferation and migration in EMOs. By taking the advantage of this scaffold-free endometrial organoids, they revealed new mechanism of PCOS-associated risk of endometrial neoplasia. So what is the future directions on endometrial organoids? First is the ability to genetically modify the cells. Second is to screen for markers of endometrial diseases and identify novel therapeutic targets. But we cannot ignore challenges on endometrial organoids, and these challenges need us to research more. Are you interested in endometrial organoids? Thank you for your listening.